Hello listeners, I am Chandana Srisai, a postgraduate student from Department of Dairy Science and Food Technology, Banaras Hindu University. This lecture is to study about the clinical testing of nutraceuticals and health foods. Let's start with the introduction of nutraceuticals and health foods. The term nutraceuticals is a combination of nutrition and pharmaceutical coined in 1989 by Stephen DeFelice, who is the MD, founder, and chairman of the Foundation for Innovation in Medicine. Nutraceutical product can be defined as any substance that may be considered a food or product of a food that provides medical or health benefits, including the prevention and treatment of disease. Nutraceuticals are the isolated, purified, and formulated healthful products which are taken in the form of capsules, tinctures, or pills or tablets. Health foods can be defined as foods or dietary components that may provide a health benefit beyond basic nutrition. They complement the regular diet, provide nourishment for the body, and fight disease. They provide all necessary nutrients, keep us fit, and add to our overall well being. Health foods not only provide complete nourishment but also have healing and rejuvenating properties. They can be in the form of herbs, cereals, fruits, or any other blend of natural ingredients. The examples can be spirulina, ginseng, wheatgrass, etc. The health and fitness consciousness of consumers, readiness of people to pay for health foods and additives resulted in rapidly growing the popularity and growth of nutraceuticals and functional foods in the market. The Indian nutraceuticals market can be categorized broadly into functional foods, that is 68%, and dietary supplements, that is 32%. Nutraceuticals and health foods play a unique role in contributing to health and protecting against illnesses outside the traditional scope of drugs by adding on to the basic nutritional value consumed in food. Consumers use it as an attempt to accomplish desirable therapeutic outcomes with reduced side effects. However, with all of the aforementioned positive points, nutraceuticals still need support of an extensive scientific study to prove their effects with reduced side effects. The exponential growth in the nutraceutical product and health foods requires clinical trials to support safety and efficacy. Now, trials can help sponsors to meet these requirements by providing proof of a nutraceutical's performance. And also, a clinical trial evaluates the safety, efficacy, and bioavailability of nutraceuticals, the latter of which measures the extent and rate at which the nutraceutical is absorbed into the body. The focus of a trial can include disease modification, disease recovery, etc. Now let's study about the clinical research. Let's have a glance about the history of clinical research regulations in India. India has been an attractive market for clinical trials for decades owing to the diverse large population and favorable regulatory landscape. The history of clinical research regulation in India can be tracked back to the 1940 Drugs and Cosmetics Act. It is the India's primary drug control legislation administered by the Central Drug Standard Control Organization. Originally, this only dealt with import, manufacture, distribution, and sales of drugs in India, and clinical research was not regulated. In 1970, the colonial patent laws were replaced by the Indian Patents Act 1970 to create a process patenting regime and boost domestic production. As a result, India became a hub for generic drugs and foreign pharmaceutical manufacturers from entering the Indian market. Subsequently, in 1988, Schedule Y was introduced in the DCR, that is Domestic Cosmetic Rules, to regulate clinical trials in India to support the growth of a predominantly generic Indian pharmaceutical industry. This amendment created a sound regularity regime of clinical trials by introducing a four-phase clinical trial scheme and legalized the guidelines on good clinical practice in India in 2001. Parallelly, the Indian Council of Medical Research (ICMR), India's apex regulatory body for biomedical research, came up with the guidelines for biomedical and health research involving human participants, along with Schedule Y of the DCR, framed the clinical trial regulatory framework in India. Presently, the CT rules along with CDC-COGCP frame the clinical trial law, and ICMR guidelines for biomedical and health research involving human participants, issued in 2019, frame the clinical research law in India. Human subject and volunteers or patients are an important consideration in clinical trial. There are certain factors influencing the selection of volunteers or patients for inclusion or exclusion from the trial. Pregnant women, children, seriously ill patients are generally excluded from clinical trials unless the study is designed specifically to them. Patients having risk of an adverse reaction would also be excluded. Clinical trials are usually carried in younger patients because certain products are found to be more allergic in elderly patients. The selected subjects must be assigned to the drug treatment or control group in an unbiased manner, which is usually done in a random assignment. This assignment is known as experimental design for clinical trial, which is of two types. First one is randomization, which is to avoid the selection of volunteers or patients who are different in some respect. Randomization is necessary. The order of giving certain treatment group allocated to the subjects should be decided before the trial starts. Each volunteer or patient is coded with certain code number so that investigator knows that which treatment an individual is receiving. The second one is a controlled clinical trial, that is, when a new drug therapy is compared with a previously established therapy or placebo under standard conditions, it is called the controlled clinical trial. The studies are conducted in two ways. Firstly, one drug is given to the first group and the result will be compared with the second group. And secondly, the one drug therapy is altered with control therapy or with previously established drug therapy. And the controlled trials are again of two types, that is blindness and placebos. Discussing about the blindness, in a clinical trial, blindness means that volunteers and doctor do not know what treatment is being administered, and it is done to eliminate the bias. The blindness in trial is of again two types, that is single blind and double blind. Single blind is where the patient does not know what treatment he is receiving, and the double blind is neither the patient nor the doctor knows what treatment the patient is receiving. In this technique, the drugs or either the capsules or tablets or injection are coded. Coming to the placebo, placebos are used in double blind trials. They are made to match the active treatment with respect to the size, color, taste, smell, texture, and weight, etc. Example: lactose capsules, saline injections, etc. They are pharmacologically inert. This is a clinical trial life cycle where we have the first step is drug discovery, and the second one is preclinical research, and the third step is clinical research. Fourth is safety testing in animals and trials in humans. Coming to the first, drug discovery and development. This has three sections where first the scientists carry out basic research in chemistry, biochemistry, physiology, microbiology, and pharmacology to understand natural substances and physiological processes associated with the purpose of drug development. The data gathered during the research and discovery phase is translated into disease-specific potential new drugs. And coming to the screening, this is the stage of where the chemical compounds are screened in order to observe their pharmacological effects. Rapid high efficacy drugs screening facilitates the determination of whether a chemical compound has characteristics of a potential drug for the disease intended to be cured. Then the last step is the identification, where the efficacy of a chemical compound is identified at this stage. This is done using in vitro screening techniques. Coming to the second step, that is preclinical researches. This in vitro testing is followed by in vivo testing. Both these types of testing form part of good laboratory practices, along with pharmacodynamics, pharmacokinetics, etc. The preclinical stage is crucial as it is the first safety valve before in vitro testing and helps in determining the mechanism of action for the lead compounds. The aim should be to ensure sufficient safety and efficiency, which is a prerequisite for regulatory authorities to approve the progression of to the clinical phase of a new drug. The schedule of the preclinical research in relation to clinical trial must be decided upon by taking into consideration the characteristics of the new drug, the disease which it is intended for, the duration and exposure of the clinical trial subjected to the drug and the route of the administration. Coming to the phases of clinical trials, we have four phases where the phase 1 is a human pharmacology, phase 2 is a therapeutic exploratory, phase 3 is exploratory analysis, and phase 4 is post-marketing surveillance. We would discuss about each phase detailedly. The first phase, human exploratory phase, the objective is to test the safety and dosage of the drug, especially with regard to the level at which toxicity first occurs, as opposed to being undertaken with a therapeutic objective. At this preliminary stage, the capacity of the drug to reach the target organ or site of action to prevent a condition and the human body's tolerability towards a drug is observed. The subjects may include about 20 to 100 people, either healthy or certain types of patients. This phase can last for several months. It should be carried out by investigators trained in clinical pharmacology. It may include early measurement of drug activity by undertaking pharmacodynamics and pharmacokinetic studies. Phase 2 is a therapeutic exploratory phase where the purpose of phase 2 is to check the therapeutic efficacy, spot the side effects, and concretize a dosage pattern of the drug. Up to several hundred patients with the disease volunteer as trial subjects. The set of subjects is relatively homogeneous as it is selected through narrow criteria. The subjects are divided into two sets, one under the influence of the new drug and the other an old drug or an older version. The volunteers are deprived of the knowledge of which set they are in so as to prevent them from developing a bias. This process is known as blinding. Phase 2 can last for several months to 2 years till the dose and regimen of phase 3 is finalized. Phase 3 is the exploratory analysis. It is a large-scale version of phase 3 trials. The efficacy of the drug and the adverse reactions of the target human population to the drug are observed and noted to confirm the therapeutic benefits. Along with this, dosing levels are confirmed and a harmful benefit analysis is conducted. Several hundred to several thousand patients who are likely to use the new drug are the subjects. This creates the requisite basis for the new drug to be used firstly in wider population, secondly at different stages of the disease, and thirdly the safety of the drug in combination with other drugs may be determined. Although extended exposure to the drug may be initiated in phase 2, it is arrived at in phase 3 to complete the drug prescription. This phase goes on for about 1 to 4 years and may include studies in special populations such as children, pregnant women, the elderly, and patients with organ system failures, etc. The methodology used in this phase is exploratory analysis, and the safety in comparison with existing approved drugs is examined. fourth phase is a post-marketing surveillance, which must be conducted for each new drug approved for marketing in order to take cognizance of all rare adverse events leading from the utilization and application of the drug in a larger population. Once a drug has been cleared by the regulator, it is made available to patients either with a prescription or over the counter. As a drug starts being used widely, data is gathered in order to enhance the understanding of its efficiency in different circumstances during the lifetime of the medicine. This propels gradual developments. Coming to the safety testing in animals, it is performed in animals to select the most suitable lead and the most efficient form of dosage. The final step is the approvals for inhuman trials. Drug regulators regulate the development and marketing of medical interventions for ensuring suitability and safety for the public. Therefore, to administer drugs for the purpose of research, the clinical trials also require prior approvals and the trials scrutinized and monitored by the drug regulators. Coming to the laws and regulations, the Indian legal framework and regulation under which clinical trials and medical research have to be carried out mainly comprises of the following. The first one is Drug and Cosmetics Act 1940 and Drugs and Cosmetics Rules 1945. The second one is New Drugs and Clinical Trials Rules 2019. National Ethical Guidelines for Biomedical and Health Research Involving Human Participants 2017, Good Clinical Practice Guidelines for Clinical Research in India issued by CDSCO, Good Clinical Laboratory Practices. There are various regulating bodies across the world. In India, ICMR, Canada, Health Canada, UK, Medicines and Healthcare Products Regulatory Agency, USA, FDA, EU, European Medicines Agency, Japan Pharmaceutical and Medical Devices Agency. We will look into a case study. The case study here shows the clinical trials of 5 different food supplements with restriction diet in adults. This study is designed as parallel randomized double-blind study exploring the efficacy of 2 months daily oral dosing of 5 different food supplements together with diet restriction in 120 otherwise healthy overweight or obese adults on different biochemical uh, and anthropometric parameters. Food supplements are mainly composed of different fiber and other bioactive compounds. Here are the references used in the lecture. Thank you.